I am Dr. Atma Selvi, Assistant Professor in the Department of Food Process Engineering at SRM University, Chennai. Dear students, in this lecture, I will discuss about concentration of liquid food by freeze concentration and membrane concentration. As we all know, heat is used as the most common method of liquid food concentration. Water activity of the liquid food is reduced due to the loss of moisture during evaporation. But this method of liquid food concentration results in loss of sensory and nutritional properties of the food. Hence, there is a need for alternative method of liquid food concentration. In today's lecture, I will discuss about concentration of liquid food by freeze concentration and membrane concentration. The objective of this lecture is to understand the concentration of liquid foods by freeze concentration, to understand the concentration of liquid food by membrane concentration. Freeze concentration has been considered as an efficient method. In this method, water is removed as ice rather than as vapor. In freeze concentration, the product is cooled below its freezing point, allowing water to form crystals, which are then removed from the concentrate by centrifugation. Freeze concentration selectively removes water from the food without altering any other components. The preservation effect of freeze concentration is due to the reduction in the water activity without the application of heat. The low temperature used in the process cause a high retention of volatile aroma compounds, as a result of which nutritional qualities and sensory characteristics are better maintained. Therefore, the concentrated product gets higher quality than that processed by thermal evaporation. Now, let us see the theory of freeze concentration. When a solid or a liquid food is frozen, the components present in the food will not freeze at once. First, the water gets frozen, forming ice crystals in the mixture. The remaining unfrozen food solution is now high in solids concentration. Before the entire mixture freezes, it is possible to separate the initially formed ice crystals. This separation could be done using centrifugal separation or filter press. The concentrated unfrozen food solution is again passed through series of screens, while the frozen water crystals are retained and can be discarded. Repeating this process several times, concentration of the unfrozen food solution can be increased several fold. Now, let us discuss in detail the steps involved in freeze concentration. The first step is fractional crystallization of water to ice, and the next step is the growth of ice crystals, and finally the removal of ice from the unfrozen solution. Fractional crystallization of water to ice is done using direct contact freezing system like solid carbon dioxide or liquid nitrogen, or using indirect equipment to freeze the liquid foods. Growth of ice crystals. The ice crystals formed are slowly agitated in a mixing vessel such that it grows to bigger size. Large crystals generally get separated from the concentrate easily. In freeze concentration, it is preferred to grow the ice crystals bigger in order to reduce the amount of concentrated liquid entrained in the crystals. Growth of ice crystals. The ice crystals formed are slowly agitated in a mixing vessel such that it grows to bigger size. Large crystals generally get separated from the concentrate easily. In freeze concentration, it is preferred to grow the ice crystals bigger in order to reduce the amount of concentrated liquid entrained in the crystals. It is done by slowly stirring the slurry, that is the mixture of ice crystals and liquid, and permitting the large crystals to grow. Next is separation of crystals from the concentrated solution. Separation of crystal so formed due to the conversion of water to ice is achieved using gravity drainage, filtration, filter pressing, centrifugation, or wash columns. Wash column functions by feeding the ice concentrated slurry into the bottom of a vertical cylinder. The greater part of the concentrate passes through the crystals and is separated. Heater is placed at the top of the column to melt the formed ice crystals, and the portion of the melted water passes through the bed of ice crystals to remove entrained concentrate. Concentration is done in single stage or more. Generally, multi-stage equipments are used. A multi-stage concentrating system consumes less energy and has higher production rates. Advancement in the technique for generating bigger crystals and more efficient washing has increased the maximum obtainable concentration to 45% solids. The efficiency of separation of crystals from the concentrated liquor is determined by the degree of clumping of the crystals and the amount of liquor entrained. Now, let us see the advantages of freeze concentration. This process can be carried out at low temperature, high retention of volatile aroma. The disadvantage of freeze concentration are: the concentration achieved by freeze concentration is lower than the values obtained by evaporation. Separation of smaller ice crystals formed during rapid freezing is difficult. Freeze concentration is not suitable for highly pulped liquids. High refrigeration cost, high equipment investment to handle frozen mass, and high operating cost. Freeze concentration has low production rate than conventional dehydration, evaporation, or membrane concentration. Due to these reasons, freeze concentration is only used for high value product. So far, we have learnt about concentration of liquid foods by freeze concentration. Now, let us discuss about concentration of liquid food using membrane. As I already discussed, evaporative concentration of fluid food deteriorates the sensory properties like color, flavor, and taste of the foods, and it also deteriorates the nutritive value of the food. In membrane concentration process, most of the sensory properties and nutritional properties of the food are retained. Membranes are material that have voids in them, allowing some molecules to pass more conveniently and retain some other molecules. So, in membrane separation process, a membrane is placed between two phases, that is, feed and permeate. In membrane separation system, a fluid containing two or more components is in contact with a membrane that permits selected components, for example, water, in the fluid to permeate more readily than other components. One of the earliest successful industrial application of membrane technology was in the food industry. In 1972, a dairy plant in New York began processing cheese whey by reverse osmosis. Some of the application of the membrane separation process in food industry includes purification of water, concentration and clarification of fruit juices, milk products, alcoholic beverages and food industry waste water. In order to understand the principle behind the membrane-based separation, we need to understand the principle of osmosis. Osmosis is a naturally occurring phenomena and one of the most important processes in nature. It is a process where a lower concentrated solution will tend to migrate to a strong concentrated solution, as the same way plant root absorbs water from soil. Once a concentration in both chambers have reached equilibrium, the movement stops as enough pressure has built up to stop the flow of water from one chamber to the other. This pressure is known as osmotic pressure. Now let us see about reverse osmosis. As we know, osmosis is naturally occurring process. Energy need to be supplied to reverse the process of osmosis. Reverse osmosis is a process in which pressure greater than the natural osmotic pressure is applied on the high concentration side of the membrane, forcing the water to travel through the membrane from the higher concentration to low concentration, thus reversing the natural tendency of water flow. Reverse osmosis requires high pumping pressure to push the water through the membrane. Membrane separation are classified by pore size as microfiltration, ultrafiltration, nanofiltration, reverse osmosis and forward osmosis. If we look at the spectrum, membrane pore size and the applied pressure difference are often used to distinguish the processes from each other. The only problem in membrane separation is that during this concentration process, on the retentate side of the membrane, the concentration of the total soluble solids becomes higher and higher, causing higher osmotic pressure and decreasing the driving force of concentration process. Ultrafiltration membranes have a higher porosity and retains large molecules, for example, protein or colloids, which have a lower osmotic pressure. Smaller solutes are transported across the membrane with the water. Ultrafiltration membrane have a porosity of 1 to 100 nanometer. Ultrafiltration therefore operates at low pressure of 0.01 to 0.055 megapascal. Ultrafiltration can be carried out at room temperature or in a cold room. Main application of ultrafiltration in food industry is the concentration of milk, recovery of whey proteins, recovery of potato starch and proteins, concentration of egg products, clarification of fruit juices and alcoholic beverages. Ultrafiltration technique is also used in pharmaceutical industry, chemical industry, textile industry and paper industry. Next, let us see about microfiltration. Microfiltration is the method of removing particles of size 0.025 micrometer to 10 micrometer. In this, the membranes have pore diameter ranging from 0.1 to 10 micrometer. The hydrostatic pressure difference used are in the range of 0.01 to 0.1 megapascal. The liquid is pumped in from the outside of the membrane, and from inside of the membrane the clean water is collected. A filter cake that deposit on the membrane is need to be removed by back washing. Recovery rates are more than 90% with feed sources having low turbidity. This method is useful for harvesting microbial cells from fermentation broth. Membranes can be used for separating constituents of food on a molecular basis, and thus for a membrane to work efficiently, we require to understand all the ideal conditions required for a membrane. The optimum condition for a membrane are: it should offer high degree of separation, it should withstand high pressure, membrane should be operational at moderate temperatures, it should work at low pH ranges, membrane should have resist microbial effect, and the membrane should be cost effective. Majority of the membranes are made up of cellulose acetate, polyamides, polysulfones, polyacrylonitrile, polypropylene and inorganic ceramic. The operational temperature range for cellulose acetate should be less than 30 degrees centigrade. For polysulfones and polyamides, the operating temperature should be less than 80 degrees centigrade. And for ceramic, it should be less than 130 degrees centigrade. The pH range of cellulose acetate is 3 to 8, and for polysulfones it is 3 to 11. The average thickness of the membrane should be 0.5 to 1 micrometer. Membrane modules used in microfiltration are plate and frame module, which is used in filtering dilute solutions, and spiral wound module, which is preferred when the solutions handle heavily concentrated solution and in conditions of high temperature and extreme pH. Now let us discuss about nanofiltration. Nanofiltration membranes have pore size of approximately 0.001 microns. Nanofiltration requires high operating pressure than either microfiltration or ultrafiltration. These systems can remove virtually all cysts, bacteria, virus and humic materials. There are two types of membranes used in separation. Spiral membranes, which is more reactive to pollution, and tubular membranes, which is not easily prone to pollution. Some of the applications of nanofiltration include color removal, removal of low molecular weight species such as organics in pharmaceutical production, surfactants from waste water. In food industries, nanofilters are used in demineralization of whey, fruit juice concentration, molasses purification for reuse, and in wastewater treatment. So far, we have learned about ultrafiltration, microfiltration, and nanofiltration. Next, let us discuss about forward osmosis. Forward osmosis, also known as direct osmosis, is a promising membrane technology, and its only driving force is a difference in osmotic pressure. The transfer of water occurs from low concentration side to high concentration side across a semi-permeable membrane, and this process continues until osmotic pressure difference between the sides are nil. Here, osmotic pressure difference is the only driving force for the movement of water. Now, let us see the advantage of forward osmosis. Low hydraulic pressure for operation, which reduces the cost for electrical energy. Low processing temperature, which avoids the thermal degradation of food quality factors. High product recovery and low discharge of brine to the environment. Low irreversible fouling, which leads to low cost on membrane cleaning and replacement. Efficient treatment of feeds with high solid content. A hydrophobic membrane exhibits a barrier for the liquid phase and allows only the vapor phase to pass through the membrane pores. The driving force for this process is provided by partial vapor pressure difference caused by temperature difference. The surface of the membrane are in direct contact with two liquid phases which are kept at different temperature. The feed, which is a warm solution, and the permeate, a cold solution. Vapor is transported through the membrane as gaseous phase exists inside the membrane, and the movement of the vapor continues until the pressure difference is maintained. Now let us discuss about vacuum membrane distillation. Vacuum membrane distillation is a membrane separation process that removes volatile compounds from the aqueous solution. In this, one side of the membrane is passed with the feed, whereas the other side is vacuumed. The membrane is hydrophobic and microporous, and so the solution in the feed side do not pass through the membrane towards the vacuum side due to interface tension. When the pressure of the vacuum side is kept below the equilibrium vapor pressure of the solution, the water present in the feed side gets evaporated and moved towards the vacuum side. Meanwhile, the vapor entering the vacuum side is vacuumized outside the membrane cell. Finally, let us see about dialysis or liquid permeation. Dialysis is a membrane process where dissolved substances that has a molecular weight less than 100 Dalton diffuses from one side of the membrane, that is feed side, to the other side, that is permeate side, according to its concentration gradient. The solutes separate by passing through the membrane that act as fiber filter, and the separation occurs by a screening action based on the pore diameter and particle size. In this case, the small solutes in one liquid phase penetrate through a porous membrane to another liquid phase where the permeates are diluted by means of a sweeping solution. The permeates can be separated by flushing from the sweeping liquid when its boiling point is much lower than that of sweeping liquid. This dialysis process is called as petrification. In practice, dialysis is used in separating microbial species that differ in size, which have large difference in diffusion rates. With the use of dialysis, sodium hydroxide in cellulose processing can be removed, acid can be removed from metallurgical liquors, removal of product from a culture solution in fermentation, and reduction of alcoholic content of beer. Other therapeutic application includes dialysis in removing toxic substances such as urea or creatinine from the blood of patient with chronic uremia. Advantages of membrane concentration. The advantages of membrane concentration over concentration by evaporation, or primarily the food is not heated and there is therefore negligible loss of volatiles or changes nutritional or eating quality. In contrast with boiling, membrane concentration do not involve a change in phase and therefore uses energy more efficiently. In addition, it's simple installation with low labor and operating cost and requires no requirement for steam boilers. While the main limitation of membrane concentration are variation in the product flow rate when changes occur in the concentration of feed liquor, higher capital cost than evaporators, a maximum concentration achieved is 30% total soluble solids, fouling of the membranes, that is deposition of polymers, which reduces the operating time between membrane cleaning. Let me summarize this lecture. Freezing is an energy efficient process that can concentrate food liquids containing volatile aromas without any appreciable loss of quality. In the past, the process was not much used because of high investment cost and the considerable loss of concentration in the withdrawal ice. New technological developments have minimized these two hindrances associated with the freeze concentration process. Membranes are materials that have voids in them, allowing some molecules to pass more conveniently than other molecules. Osmosis is a process where lower concentrated solution will tend to migrate to strong concentrated solution. Process continues until equilibrium is reached. Reverse osmosis is a process in which pressure greater than natural osmotic pressure is applied on high concentration side of the membrane, forcing the water to travel through the membrane from the high concentration to low concentration. Membrane separations are classified by pore size as microfiltration, ultrafiltration, nanofiltration. Reverse osmosis and ultrafiltration are seen as attractive and very competitive technique for dewatering of liquid foods such as milk and whey. The only problem in membrane separation is that during this concentration process, on the retentate side of the membrane, the concentration of the total soluble solids become higher and higher, causing higher osmotic pressure and decreasing the driving force of the concentration process.